Was that the sound you get when I start the recording? I'll be doggone. All right, essay question. Let's see. I got two of them for you. Both of them are the same type, compare and contrast. Yeah, now, you guys understand what compare and contrast questions are, right? We talked about that before. Compare and contrast questions is going to be, I want you to tell me how the things are similar and how they're different, all right? So, for example, if I were to ask you to compare and contrast mitosis and meiosis, you want to tell me about the different stages and how some stages occur in meiosis and don't occur in mitosis. So those slides that I showed you side by side, those are really good things to study when it comes to that. The other one is another compare and contrast, and it's going to be respiration and photosynthesis. So as you're going through and studying those things, make sure you understand the similarities between them, but make sure you don't confuse them and begin to blend them together, all right? So that's going to be your two essay questions. Compare and contrast mitosis and meiosis, and then do the same thing for my respiration and photosynthesis.
Are we picking one or the other?
Penny asks, Penny asks, can we pick one or the other? No, you're going to do both of them. I'm going to have you do both of them, okay? I know it always seems that way, but honestly, in the past grading your exams, it seems like y'all do better on the essays than you do on the rest of it. So this is 40 points of essays, so that's almost half the class, half the exam. So I'm hoping to get some good results out of that. Any other questions about the exam before I kind of go off with chapter 14? I know we started it, but I do want to step back a little bit again because I always like to pick up and overlap a little bit. Are we all good out there? Penny, you're good?
Yes, coming.
All right, excellent. All right, looks like everybody's online. We're all set. Recording's on. Okay, so once again in chapter 14, as I said, what we've done is we're drilling down now to actually the molecular level. So we're moving beyond the idea that there's dominant alleles and recessive alleles and all that stuff we talked about, and we're kind of doing a full circle thing, believe it or not. We're kind of going back to where we were, or gosh, I don't know, chapter 3 or 5 or something like that, when we actually talked about the different types of organic molecules, right? We talked about proteins, and we talked about lipids. Yep. Yeah, we are recording. Thank you, Emily. We talked about lipids, and we talked about proteins, and we talked about carbohydrates, and we also talked about nucleic acids at that time. Didn't spend a whole lot of time on it. Gave you a few little ideas about how the DNA molecule looks. You know, I introduced you to the idea of the base pairing, that adenines bind to thymine and all that kind of stuff. And I even hinted back then, I said, okay, that sequence of letters, folks, that's a code. And we're going to eventually learn how we're going to read that code. Now, we haven't gotten quite to that point where we're going to be able to read the code, but nonetheless, we do know there is a code. We get into subsequent chapters, and I'll show you how we actually do read the code. So that's what we're doing. We're getting down to the molecular level of how this DNA information, how that sequence of letters turns into a sequence of proteins. And a sequence of proteins, you remember, was the primary level of protein structure, right? And we talked about proteins, 4 levels. The first one was the order of the amino acids. And as a matter of fact, some of the early work on heredity, in other words, what was it in the cell that was inherited, we thought it was a protein. We didn't know about nucleic acids at that time, so we thought it was a protein, and they did a couple of things. One of the things very simple they did is they took an enzyme that digests proteins, and they treated cells with it. And the idea is if what's in the nucleus is going to...
I'm shutting up.
Thank you. I'm hearing my... Penny's got her mic on. It's like being in an echo chamber. Thank you, Penny. But if you think about that order of amino acids that we were looking at, that is a code as well, right? I mean, there's 20 different amino acids, and you can arrange them in different ways, and we know that affects the structure. But the recipe to put those together, and I even said this back then, I know I did, was I said the recipe of that code is something we'll learn later on in the semester. So that's kind of what we're getting to right now. We're getting ready to turn that recipe that's in your DNA into a sequence of amino acids, which, of course, makes a protein. But first, we've got to give credit where credit is due. And so let's do a little history lesson and talk about some of the folks that helped us get where we are in terms of what we knew about DNA. This is actually kind of neat because DNA was only discovered, or not discovered, it was only figured out the way it looks back in the early 50s. So it wasn't much right before I was born, which is kind of neat if you think about that. Science is always changing, and something that you guys, younger folks, take for granted is we always knew Right? Everybody knows about DNA and talks about DNA, but back in the 50s, it was something that was being debated, so it wasn't that long ago. But if we jump back even further than that, we go back to Miescher, and there's a picture of him right there in the 1860s. When we were looking for the genetic material, common sense might have told you that perhaps that genetic material was in the nucleus. And there's a couple of reasons why I'd say common sense would tell you that. If you remember looking at a cell under the microscope, there is the cell wall, if it's a plant cell, then there's the cell membrane, and then there's another membrane around the nucleus, and there's stuff inside of there. And the genetic material, as you imagine, is pretty important. It's your code, right? So you are. So right away you're going, there's something in there that's being, really protected. It's being protected inside the membrane of the cell and in the membrane of the nucleus. And no doubt those folks, when they were looking under the microscope, might have seen things similar to what you see here on the right. As I'm trying to draw with my pen that doesn't work. Thank you, Courtney. As you see on the right-hand side, no doubt when they were looking at these cells, and especially if you jump up to Fulgen there in the 1900s, when they began to stain these cells, we got to be able to follow these chromosomes around, essentially. So you can imagine that someone looking through a microscope back then might have been able to see those cells going through mitosis like you did and went, huh, that's interesting. We all know cells have to have that code. We all know they have to have that DNA. And when we look at mitosis under a scope, you can see that it looks like something's being duplicated and then divided. So wow, that might be the genetic code, because each cell seems to have a copy of whatever those little purple squiggly things were that we're seeing underneath the microscope there. So Miescher was kind of one of those folks among others that said, all right, folks, let's narrow our focus down to the nucleus. It's probably something in the nucleus that carries heredity. And then Fulgen came up with a stain. That's actually the stain that was used to color the chromosomes in the onion cells. You were looking at that was a Fulgen stain. So if you develop a stain, for years from now, they'll recall it as the Fulgen-Clark stain. They might call it something like that, right? Early 1900s. And of course, in microscopy, staining things makes them a lot more visible, a lot more easy to follow around. So now we got a better idea of looking at them. Now, Griffith in 1928, what he did is he came across something. He didn't invent this or discover that. Well, he discovered it, I guess. But bacteria do this thing called bacterial transformation. I don't talk a lot about bacteria in this class, but I do talk a lot more about it in my bio too. And one of the things we always talk about in bacteria is how do they generate diversity? We generate diversity mostly by crossing over, right? That happens in meiosis. Well, they don't do that. Prokaryotes, bacteria, don't cross over. They reproduce asexually. They just divide themselves in half, which doesn't generate a lot of genetic diversity, because when you asexually reproduce, essentially you're a clone. Essentially, all right? Every time DNA makes a copy, it doesn't mean it makes an exact copy. There's always going to be little blips along the way sometimes, but for the most part, you're going to be a clone. There's no genetic diversity there. And genetic diversity is important for organisms to survive, especially when you think about how it is for evolutionary adaptations. So what Griffith came upon was this idea of bacterial transformation. Now, we're not talking about bacteria here. We're talking about the DNA. What his experiment showed was that this molecule, or this suspected molecule, had the ability to store information. Let me skip forward a couple of slides, and I'll come back to the slide I skipped, because I got these a little bit out of order. So once again, what Griffith was doing was working with the old lab rat, and was working with a pathogenic prokaryote. Pathogenic is disease-causing, whose name is Streptococcus pneumonia. You got it. Thank you, Penny. Perfect. There it is. That's how it's spelled. It's supposed to be a capital S. Streptococcus pneumonia, which gives the disease pneumonia. Pneumonia is basically your lungs fill up with fluid, and you, not a pleasant way to go, but you basically drown from this. Now, among bacterial organisms, there are what we call strains. And you might have heard this before, for example, when they were talking about E. coli. You have E. coli in your gut, by the way. E. coli is a normal resident found in our guts. But it's the strain of E. coli that's in our guts that's okay. The E. coli that you get from food that we hear sometimes in the paper is the same organism, but a different strain. That would be what we would call a pathogenic strain, right? Something that causes disease. So Streptococcus is no different when it comes to that. In Streptococcus, there's one that's called S, and there's one that's called R. You can see in the picture of the first two, the living S and living R. Now, the reason why they're called S and R is the same reason why the rough and the smooth ER were called that way, because of the fact that they look that way. Under a microscope, living S cells look smooth, and living R cells look lumpier. And if you look at the living S cells, you see why they look smooth. That orange that's around them, that's a capsule. It's a gelatinous capsule. And what that, and the two little dots inside are the critters, or the bacteria. And what that gelatinous capsule does, and this is something that a lot of bacteria do, is it allows those S cells, those Streptococcus pneumoniae cells, to hide from the immune system of the mouse. See, your immune system only knows something is not supposed to be there in your body basically by shape and touch and feel. Obviously, your immune system can't ask, can I see your ID card, something like that. That doesn't work. So they do it by touch and feel. But if you have a gelatinous coat around you, basically you're invisible to the immune system. So with the gelatinous coat, there's no way for the immune system to fight this pathogenic strain of Streptococcus, and the poor mouse dies. That's what Griffith called his pathogenic control. In other words, he double-checked and made sure that indeed the living S kills the mouse. Then he took living R. Now you notice living R doesn't have the capsule. Living R is not hidden to the immune system. Inject that into the mouse, and the mouse immune system goes to work, assuming the mouse immune system is healthy. Mouse might get a little sick, just like you do, but you don't die. It doesn't die. It survives from it. All right, very good. Now that's the non-pathogenic control. So he set up the idea that the difference between these two strains, think of strains maybe as breeds of dog, right? They're all the same species, but there's different breeds of dogs. And you might even extend that to say some breeds are a little more dangerous than others, right? It's kind of like nobody's worried about a Yorkshire Terrier, taking you down. Maybe a couple hundred of them. But then there are larger, more powerful dogs that certainly could do something like that. So this living S and living R, just two different strains. And the living S is the disease-causing one, and the living R is not. Now, he then goes to the non-pathogenic control in the third setup there, where he takes the S and he kills it, boils it, puts it in pressure. In other words, kills the organism, destroys that gelatinous capsule that's around that, and injects that into the mouse, and the mouse remains healthy. Okay, well, why would he do that? Well, sometimes your body responds to things and makes yourself sick in response to, oh gosh, kind of went off on the wrong, in response to what your body has taken in. Let me see what I mean by that. In the third one, what Griffith was trying to figure out was it the living S that kills the mouse, or could it be the mouse's immune system goes overboard at the living S and kills the mouse? Peanut butter. Folks who are allergic to peanut butter. There are some people that are so allergic to peanut butter that just the smell of it can put them into anaphylactic shock. An anaphylactic shock is your immune system gone haywire. What it does is it closes up the air passages in your lungs. It makes it hard to breathe, and you literally could die from being exposed to peanut butter, right? Even if you don't have allergies that bad, when it's allergy season, your body feels like it's got a cold, right? The nose runs and you cough and you sneeze, but you don't have a cold. Your body's acting like it's got a cold. It's treating that tree pollen, which would never make you sick in a million years anyway, but it's treating that as something foreign. It's going, I don't know what this is, but I need to get it out of the body. So the nose runs and you cough and sneeze because it's trying to rid itself. And you might even get a fever too. A fever is a response by organisms because if they jack up the temperature, then the organism that's inside, the prokaryotic, the bacteria, might die, might kill it off. That's what a fever is for. So in that third one, he demonstrated that sure enough, it's not the mouse's immune system that killed the mouse in the first one. It was indeed the living S cells, because things that are dead can't kill you. Right? He killed in this particular case. Now, here's in the last one, and this is where he got his information about how this molecule has the ability to store information. He puts a mixture of the S that he's killed and the living R that he injected in the mouse before, and he injects into the mouse, and the mouse dies. Now, shouldn't have died, right? He killed S, didn't kill it. Living R didn't kill it. But if I put them together, the mouse dies. And then when he did a necropsy on the mouse, not an autopsy, you can't do an autopsy unless you're a mouse, right? He did a necropsy in the mouse, and he found inside the mouse living S. Now, he didn't inject living S into the mouse, so that's a puzzle, right? Well, what he had found out was this bacterial transformation thing I was talking about. Bacteria have the ability, this is one of the ways they generate genetic diversity, they have the ability to take up DNA from the environment, even across species lines, even from another species. Now, I know, I'm pretty sure that has to stay within the prokaryote species, but if there's a dead cell and DNA is leaked out of that cell, then the prokaryotic organism, many bacteria, can take up that DNA and incorporate it into their own DNA. And now when they got it in their own DNA, they have information. So what was going on, and the reason why the mouse died in the last one, is when you're dead, your DNA is still there, right? I mean, entropy, eventually it falls apart, but right after you die, you know, grandma laying there in the casket, her DNA is still there. If we could extract her DNA, we would still have all the information about what made grandma grandma. So when those heat-killed S cells died and all their DNA was spread out into the environment, the living R cells took it up, right? They bacterially transformed themselves. They incorporated that DNA, and in that DNA was the information on how to make the capsule, right? And so now, once those living R cells know how to make a capsule, they're no longer living R cells. They're now living S cells. And that's what kills the mouse. This here demonstrates very convincingly that this molecule we have has the ability to store information, because the information that was in the S was used by the R. And if you remember, I said that the DNA molecule being able to store information, that's a little unusual. Molecules don't store information, right? Like, for example, this molecule here. I'm writing with my finger. What is that?
CO2.
Yeah, CO2. Carbon dioxide, right? It's just a molecule.
Mm-hmm.
Does it store any information? No, it's just a molecule. It's like water, right? DNA is unique, of course, because it does store information. So here is research work that was done that showed us that there is this ability to transfer information between these species, so that this molecule that we're looking at, you know, again, we have another tick mark in the column that says maybe this is the hereditary molecule. Nothing is discovered. You know, those eureka moments where we think about, you wake up in the middle of the night and go, I've got it, eureka, those don't usually happen. Those don't usually happen. It's usually peeling away a layer of an onion, a little piece at a time, and maybe you get a few lucky eureka-ish moments along the way, and finally you come up with the answer. So did Griffith demonstrate what DNA looked like here? No. Did he demonstrate a function of DNA? Yes. And remember, when we make a hypothesis about something, we make some assumptions. We say DNA should have the ability to store information. And sure enough, we see that in this thing with the mouse, right? Okay, I said earlier that some of the initial investigators thought that, you didn't have any questions about the mouse, I'm sorry. Sort of just kind of jumped on. No? All right, let me go back a couple slides. We'll come to that one next. Early on, the investigators, since we didn't know about nucleic acids and had looked at proteins, and they recognized that proteins were coded. I mean, you look at the different amino acids, and that's a code. So initially, the hereditary molecule they thought was a protein, right? Not a carb, not a lipid. Maybe it's a protein. So what they did is they took an enzyme that digests proteins and treated the nucleus of the cell, and they found out that all that material that Miescher and Fulgen had learned how to stain, all those little chromosomes you looked at, for the most part, they all disappeared. So, excuse me, take that back. They did not disappear. So if I used an enzyme that digests proteins, and if chromosomes are made of proteins, the enzyme should have digested them. It did not. So it's not a protein. Okay, it must be some other molecule out there, but it's not a protein, because an enzyme, a protein digesting enzyme, had no effect on it. So that makes perfect sense, right? So then Hershey and Chase in the 1950s began to use these radio-labeled phage parts. A phage is this blue thing. A phage is a virus. If Viruses come in many different forms and shapes. These are called bacteriophages, phages to eat. Bacteriophages attack and kill bacteria. So even at the bacterial level, you got things coming after you. Now, we don't talk much about viruses in this class. Really, this is kind of one of the only ways we do, because viruses they're a weird thing. They're not really alive. They're not really dead. They're kind of in between. They're organized, right? But they don't engage in metabolism. They use the host cell to do the metabolism. They evolve. They definitely evolve. But they don't really reproduce in the way we define reproduction. They do reproduce, but again, they need a host cell to do that. So just kind of don't look at this picture right away initially. A bacteriophage attacks a bacterial cell, injects DNA into that bacterial cell, viral DNA, and that viral DNA ends up in the DNA of the cell. And when the cell starts reading its DNA to crank out what it needs to be a bacterial cell, it unwittingly, because that stuff has been stuck in, it makes out viral particles too. Now, once that cell has made enough viral particles, there'll be a whole bunch inside the cell. The cell will lyse, right, split open, and those viruses will go out, and inside of them, they'll have pieces of DNA they've taken from that host cell, and they'll spread those pieces of DNA around. So that's the basis of Hershey and Chase's work. And what they were going to do, is it protein or is it a nucleic acid? Now, that little statement down below is important because that is the basis behind how they were able to separate them. There is no sulfur in DNA. If you remember back when we were looking at the structure, there was carbs and hydrogens and oxygens and phosphate, definitely a phosphate. There was adenine and guanine, but there was no sulfur. DNA does not contain sulfur. But sulfur is relatively common in proteins. Relatively common. So here's what we do on the left-hand side. First, they take a bacteriophage, and inside of that, the little brown squiggly stuff you see is a radioactive protein. What they've done is they've taken a sulfur atom, and they've made it radioactive. That's what the little number 35, that tells you that's an isotope of sulfur. It's radioactive. So radioactive, it glows, basically. So they take that phage, they let that phage attack a bacteria, the phage then injects this brown material, this protein. We're testing whether to see if it's the genetic molecule or not. They inject that into the cell, they let the cells grow for a while, they let them do their thing, then they blend them in a blender and centrifuge, and they separate them. The end result is none of the cells had that brown material inside of them. So in other words, the cells weren't copying and passing that on. Of course, in genetics, we know that's not the case, right? Right, obviously, you are a representation of a molecule, chromosomes, that were passed on to you by your parents. This quite clearly shows that that protein is not being passed on. So it's not the protein. Well, they were smart enough to say, well, if it's not the protein, reject that hypothesis. Is it nucleic acid? So let's take nucleic acid and let's let's make the phosphorus radioactive. We know there's a lot of phosphates in DNA. That was the part that connected the sugars together. Go up a couple slides. Right there. There it is. See that phosphorus? So we can take that and make that radioactive. And then again, it glows and it shows up and you can see it. So they took another phage. This time they, in the head of the phage, the red squiggly stuff, is radioactive DNA. Radioactive DNA that has been made radioactive by the phosphorus, okay? So the phage injects that, what we would call hot DNA into the bacteria. Hot because we can see it radioactively. We let these things divide. Then we blender, then we run through a centrifuge, and all of the cells after that had that red material inside of them. So you look at that and you say, okay, that's being passed on. That red material is being passed on, whereas the brown protein was not passed on, the red material was. Once again, we got, I guess you might call it circumstantial, but in science, you got the idea that you got another little check in the box that says, all right, that must be the hereditary molecule. In other words, why pass it on? We know it gets passed on, right? That's obvious. So we're backing that up, looking at that at a molecular level. All right, any questions on that? So at this point, we're going to throw out that it's a protein.
Is the 35S sulfur and the 32P phosphate?
You got it. It's a 32P is the phosphorus, and the 35S is the radioactive sulfur. Yeah, there you go. Any questions on that? All right. What we got next? Oh, Chargaff. Chargaff was a fellow who was actually a contemporary of Watson and Crick. That's a picture of Watson and Crick down here. They were a couple of interesting fellows. One was they did not like each other at all. One was an English fella. The English fella is the one standing up. You can see this is a photo op, obviously. Look, you point to something, you act like you care what he's talking about. We'll put this in the newspaper. I actually saw something pretty cool online a while back. Watson, the English fella, wrote a letter to his son right after him and Crick had figured out what the DNA molecule looked like, and he writes in a letter, "We figured it out." He makes a real quick sketch, and that's the first actual drawing anybody's ever done of the DNA molecule. For sale in some auction house for some ungodly amount of money, but I love that kind of stuff. It's kind of neat. Can you imagine getting a letter from your father? "We discovered DNA today, and then I took out the garbage." So Watson and Crick. Now, Watson and Crick really didn't do a whole lot of the work themselves. What they did is they compiled the data from everybody else. So that's kind of what I'm leading you to here. And like I said, these guys didn't get along with each other at all. One was a very staunch sort of English fella, and Crick on the left, American, he was a very much of a loose cannon and kind of just did his own thing and, that kind of stuff. So while they were brilliant scientists and they accomplished brilliant things, they weren't necessarily personal friends. And I think Crick is dead. But I think after this was all done, I don't think they ever spoke to each other again. So whatever differences they had, they were able to accomplish something. They ended up getting them the Nobel Prize right there, right? So that just means, like I tell people, "Oh, I don't like my boss, I want to work here." Many times you're going to work with somebody you don't like, right? Well, what Chargaff did, Chargaff looked at pieces of DNA. And no matter what piece of DNA he looked at, when he analyzed what was in the DNA, he found that the concentration of adenine, let me just draw one and not the other. Now, when we put brackets around things in chemistry, we mean concentration of. So the amount of adenine in a particular DNA sample, whatever that might be, is always going to equal the amount of thymine. This actually became known as Chargaff's rule, and that the concentration of guanine is always going to equal the concentration of cytosine. Now, you know why this is already, because we talked about it earlier in the semester. These things pair bond, don't they? Adenine binds to thymine, guanine binds to cytosine. It actually has to do with something very simple, as the number of hydrogen bonds. See, adenine and thymine can have 2 hydrogen bonds, and guanine and cytosine have 3. So they fit together. So adenine binds to thymine, guanine binds to cytosine. It's the basis behind why I can give you a piece of DNA and say, all right, let's go G, C, C, T, G, right? Just made those letters up, and you can tell me the other side, right? Cytosine. It's always C to G, A to T. Now, we're only talking about DNA. If you're thinking, wait a minute, what was the U, the uracil? That was an RNA. We'll come up on that later on. But it's always going to be A to T and G to C, unless a mistake occurs, and they do. This became known as Chargaff's rule. And the beauty of Chargaff's rule is if you can determine the concentration of just one of these bases, just one, based on Chargaff's rule, you can determine the concentration of all of them. Let me show you what I mean by that. You take a piece of DNA, right? You analyze it. You analyze it for the concentration of adenine, let's say. I could pick any one of these, but we'll just say adenine. So in my sample of DNA, I have 10% adenine. So that's how much A's I have. 10%. I'm not giving you a number, I'm giving you a percentage, but it works the same way, right? What percent thymine do I have? Why 9? I don't know, we aren't to 100 yet. We still got guanine and cytosine to go through. Yeah. 10% adenine? Anybody? If I have 10%, I got 10% thymine. There you go, right? Because they match. For every A, there's a T. Okay, so we got 10% adenine, 10% thymine. That's cool. We're halfway there. All we need to know now is the cytosine and the guanine. Well, you know everything has to add up to 100%. Here's 20% of it right there, right? So the guanine and the cytosine must make up the other, what, 80%? And then specifically, what would be the concentration for guanine and cytosine then? If both of them together, yeah, there we go. Beautiful. 40% would be guanine. 40% would be cytosine. That's kind of cool, isn't it?
Yeah.
As long as you know one, and you know they have to have equal concentrations, just a percentage of one, I can give you the percentage of all the others. All right, and of course what you know this is doing is this is the rungs of the ladder, the DNA. This is the rungs of the ladder, that double helix molecule that holds it together. And more importantly, of course, this is your code. The sequence of letters is your genetic code, okay? So now with all of this information, including the work that was done, and let me go back a little bit, by a male and female team named Wilkins and Franklin. Here she is. This is Rosalind Franklin. Brilliant molecular biologist. Died early in life, though. I believe it was ovarian cancer, so unfortunately she didn't live very long. But really a pioneer, especially in an era in which men dominated science. She really stuck out. I remember reading one time that one of her lab supervisors wrote an evaluation about her and said that she was a brilliant person, brilliant scientist, but that she really should be home raising children. How horrible. Right? Anyway, so Rosalind Franklin and Maurice Wilkins, her partner, they were X-raying DNA. And if you look at the picture on the right of her, this is an X-ray of a DNA molecule. Now, I know it seems a little bit weird, especially if you're not used to reading X-rays, but if you can imagine looking down the double helix of the DNA. So you're up above it, looking down straight through the middle of the DNA molecule. So you're looking this way through the molecule, right? You see that arrow? So you're looking straight down through the middle of the molecule. Now, the interesting thing about that is if you notice when you look on the X-ray, there are hits, things that obviously are there, structures that the X-rays have bounced off of. And every time that Wilkins and Franklin took a X-ray of a DNA molecule, no matter where they got it or who they got it from, those little blips that you see on that screen, and they could measure the distance between them, were always the same. There was consistency. Now, what that means to folks who are trying to figure out the structure of DNA molecule is this is going to give you a dimension. You got folks who are telling you what's in it, sugars and phosphates and adenines and guanines, and now you got these folks who are taking pictures of it and saying, all right, those components that we know fit together in a certain way. And we know that because when we X-ray them, we get the same distances each time. We get the same numbers. Now, I'll come back to those numbers when we get to the next slide, or not the next slide, but the next slide that I want to talk about. But know that every time they did that, they got these same numbers, and that helped Watson and Crick, without computer-aided design, by the way, to get down in the little laboratory there with Tinker Toys, it looks like, and glue this thing together. That's what he's pointing to, is a DNA molecule there. So you got to have what it's made up of and how it's put together. And this is no different than even building a house, right? To build a house, you got to have the materials, you got to have something, idea of how to put it together. So in the very end, they win the Nobel Prize. Their structure, I think they shared it with Chargaff, if I remember correctly, but their structure of the DNA molecule has been elucidated, it's been figured out. It's the double helix you see on the left-hand side. Double helix because a helix is a spring winding, and this has got two winding things, so it's a double helix. Those winding things, just to be a little bit more technical, that's the sugars and the phosphates. And they curl around each other. And in between those are the bases. Adenine to thymine, guanine to cytosine. All right, now, what about those dimensions? Those dimensions that Wilkins and Franklin, every time they X-ray, they got these same dimensions. Well, one of the dimensions that they always got whenever they were measuring these things on the X-ray film was this little tiny one down here in the bottom, 0.34 nanometers, right there. What 0.34 nanometers ends up becoming, when Watson and Crick put their model together, is the distance between the base pairs. As a matter of fact, when we talk about genes, how big they are, the unit we use many times is base pairs. A gene will be 500 base pairs long. So a gene would be 500 pairs of those rungs of that ladder. But the distance between each one of them is 0.34. No matter what DNA you're looking at, human, rat, whatever you're looking at, those bases are always going to be arranged the same way. It's always what we love to talk about in biology, about the unity and the diversity of life. We're all DNA, but we're all different. We all start from the same place. The 1 nanometer, and I kind of wish they would do it the other way, 2 nanometers. The molecule is 2 nanometers wide. It's 2 nanometers wide. They're showing you just half the molecule, split it down the middle, but the whole molecule from one side to the other is 2 nanometers. And they got that off of the X-ray as well. All right, now if you notice, the last value that I want to talk about is a multiple of that 0.34 times 10. Right? 3.4 times 0.34 times 10 is 3.4. So what was the 3.4 nanometers? The molecule twists, okay? And the DNA molecule goes through one small and one large twist. So it's twisting around, one smaller, tighter twist and one larger, less tight twist every 10 base pairs. So as that molecule travels through 10 base pairs, each of them being separated by 0.34, as it goes through that small twist and that large twist, that distance here has covered 10 base pairs. So the hit they were getting on the film of 3.4 was hitting on the twists. All right? Does that make sense? 10 base pairs, 3.4 nanometers is one turn of a small loop and one turn of a large loop. So that would be 3.4 nanometers to where it is there, and then they didn't finish the rest of the molecule. But once you finish this large twist here, there would be another 3.4 nanometers. Cool. Put this all together. It's there, all the other data that we have, all the other evidence we have, and voila, we got ourselves a DNA molecule. And on the far right-hand side is the space-filling molecule. It's a pretty cool looking molecule. Any questions on those dimensions? Yeah? Okay. Now, one thing I need to talk about in the middle where the word says anti-parallel. This becomes important in a couple of slides when we talk about how DNA is replicated. So let's make sure we understand what that means. The numbers that you see, the 3 and the 5, if you remember back early in the semester when we were looking at glucose, we can number the carbons. There's a number 1 carbon, a number 2 carbon. The little apostrophe means prime. So in chemistry, we say it's the number 1 prime carbon, the number 2 prime, the number 3 prime, the number 4, and the number 5. What we're looking at here, and the one on the left where it says the 5 prime end, that carbon right there is the number 5 carbon. And it's the one that's sticking out. Just the C, actually. The H's are attached to it, right? But that's the number 5 carbon, and it's the one that's at the end of the row there. If you go down to the other end where it says 3 prime end down here, the number 3 carbon, now they don't draw it, but the number 3 carbon is right there, where that OH is coming off of it. See, the 5 carbon is at the end of the chain at the top. The 3 carbon is at the end of the chain on the bottom. Okay, does that make sense to everybody? Mm-hmm. Now look at the other strand. It's reversed, isn't it? Mm-hmm. So we say that the DNA molecule is anti-parallel. Anti-parallel means 2 parallel things, and you flip one the other way. So we have a 5 prime end opposite a 3 prime end, and then at the other side, the 3 is opposite the 5. Okay, now this is not an organic chemistry class. I'm not real concerned with you being able to number carbons, but I do want you to keep that 3 and 5 prime end in your mind, because that's going to become very important when we start talking about replication here in a couple of minutes, all right? So everybody understand when I say it's anti-parallel, the 3 and the 5 prime end. Any questions on that? It's whatever carbon is at the end of the chain. If you look at the one on the right, this is the number 3 carbon, and again, this is the number 5. So they're opposite of one another. All right, let's start off with some hypotheses. Scientists love hypotheses. It's the way we work. So Messelson and Stahl, we're going to try to figure out how DNA make copies of itself. So in the picture there, you see, how would we copy something that's 5 base pairs long? Well, the suggested models that you see on the right-hand side is good science. Those are alternative hypotheses. So we've got 3 ways that we believe this can happen. One's called conservative, Semi-conservative, and then the last one is called dispersive. Let's look at conservative first. So what we're trying to do is the gray bars above represent the two sides of the DNA molecule. Don't get those confused. We're not talking about chromatids here. We're actually talking about the two strands, the two helices that are held together in the middle. Right? All the bases would occur in the middle here of DNA molecule. The conservative hypothesis says the way you make a copy of DNA is kind of the way we make a copy of the Xerox machine. You take a piece of paper, you go to the Xerox machine, you copy it, you got your copy. Now, the copy you have is all new letters, and the letters that were on the original stay there. In other words, what we're saying is it's not a mixture of the stuff that was on the original page and on the new page. What you have is a full, complete copy. Right? Okay, that's one hypothesis. I'll come back to the semi-conservative one in a minute for reasons that'll be obvious, but let's go to the dispersive. The dispersive one, boy, I tell you, parsimony, the simplest answer being the right one in science, the dispersive one already raises some issues in most folks when they look at it. Here's what the dispersive model says. The DNA molecule makes a copy by being all chopped up into pieces, and then you make copies of all those little pieces, and then you put all those pieces back together, hopefully in the same order. Wow. You can think about the chance of mistake, how that would increase with something like that, right? So we can probably throw that one out, right? And they may have even done that without even doing any tests, saying, man, I tell you, that's hard to believe with the amount of times that you copy DNA that there wouldn't be a lot of mistakes happening there, right? Now, when I was a kid and we had to do projects in school and book reports, sometimes the teachers would want you to do some illustrations, and I draw for crap. You know I do. You see me draw up on the board. So semi-conservative fits really well nicely with what I'm going to use as that analogy. One of the ways that I could increase my drawing proficiency was to trace. Right? Take a piece of tracing paper and trace over that image, and you pull away that piece of tracing paper, and I'm no Van Gogh, but it's not bad. It's a hell of a lot better than I could do on my own, right? So you look at semi-conservative replication, here's what they say happens. You take the molecule and you split it. One gray piece goes this way, one gray side of the ladder goes this way, the other gray side of the ladder goes the other way, and you use the originals as templates to build the new strain. See, the one in the blue is the new part. And if you remember Chargaff's rule, this is going to minimize mistakes. Because if you have an adenine on the old strand, like say there was an adenine right there, okay, then obviously the new strand, it's going to put a thymine, because A binds to T and G binds to C. The semi-conservative replication, kind of like tracing, minimizes your chance of making mistakes. In the end, the way Meselson and Stahl figured it out was they radiograph labeled each of those old strands, and when they copied, they found that each new strand had half radioactive and half was not. So this stuff, I'm just going to put the pen away and stop believing it'll ever work. This stuff here was radioactively labeled, the gray, and then when they let it go through replication, let it go through mitosis, this was radioactive, this was not, this was radioactive, this was not. So to them, they said, all right, so it's semi-conservative replication. That's how they figured that out. That's pretty cool, isn't it? That's what I love about being able to label stuff with all those radioactive things. You can follow it, man, wherever it goes. That's just incredible. So yes, it is semi-conservative replication. And then if we kind of go to the next step and think in the big picture, we say, is that going to minimize mistakes? Yeah. It's going to make my tracing look a lot better, even if I'm no Van Gogh, right? It's going to look a little more like something. If I tried to freehand, forget it. It would look nothing like it at all. It was horrible, okay? All right, now, these next few slides, well, this slide you do have in your book. All right, I'm going to work with this one. I don't like this picture. I changed it in my my slides after yesterday. This is the picture out of your new book, and I threw it in there to talk about something, and then yesterday when I was looking, I'm like, you know what? This is really kind of confusing. So let me, yeah, let's stay there. All right. On the bottom, the leading strand. Here we go back to the 3 to the 5 prime. When you make a copy of DNA, you have to copy it in a certain direction. You copy from the 3 prime to the 5 prime. So what I'm talking about is when you're making a copy of the DNA, whatever is doing the copying, this is the original strand, right? This one up here is the original strand. And then the strands in the middle are the new strands, right? These are the new strands. This is the new strand. This is the new strand. So we're going to stick with the leading strand a little bit longer here now. So what happens on this case is something called DNA polymerase 3, which is an enzyme. It's very cool, actually, how this works. DNA polymerase 3, with this little sliding clamp, moves down the original strand in the 3 to the 5' direction. So it starts on the 3, and it's working its way to the 5' down here. And as it does, it follows Chargas rule. So every time that DNA polymerase enzyme comes along one of these green bases, let's say it's an adenine, it attaches a thymine to it. It comes across a guanine, attaches a guanine, or a cytosine. Comes across 3 thymines, it adds 3 adenines. So it follows Chargas rule, moves down the strand with that sliding clamp, and that's cool. It's like a little machinery, man. That's so neat. And the DNA polymerase makes sure that those things bond together, and you're doing what I said. You're tracing. You're basically just following Chargas rule. The helicase enzyme that you see there in the end with the blue diamond or the blue arrow, the helicase is what unzips the DNA molecule. So it's like a boat moving through the water, and the water parts, moves across. This is my favorite one here, if for no other reason than the way you say it. Top isomerase. Top isomerase. If I took a kinked up item and I grabbed the 2 loose strands, oh, getting ready to be Christmas. So you get the light bulbs out from the garage, right? And they're just in a big mess. Here's one strand, and here's the end of the strand, and here's the plug-in part of the strand. I should just be able to pull those apart, and those things will straighten out. It's not going to work, right? They're all going to get kinked up. Spend hours doing something like that. When you unwind the DNA, when helicase is unwinding the DNA, it does the same thing and actually causes kinks to occur downstream to the right there that you see. Now, those kinks are obviously going to stress the molecule and eventually break it. We don't want that to happen. So what topoisomerase does is it unbinds the DNA, unwinds the kink, and then binds it back together again. So it keeps it from kinking up as that helicase is pulling those pieces apart. Isn't that a neat little enzyme, man? So that's exactly what it does. It breaks the DNA at the phosphate bonds when they're kinked, and it breaks them and lets them unkink and then puts them back together. Kind of like if you were taking a garden hose. That'd be another one. If you grab the garden hose and you go running on the backyard, especially if the garden hose is on, you're not going to get very far until it's kinked up somewhere. So topoisomerase takes care of that, okay? Now, the RNA primer and the primase. This is kind of interesting. We think probably the first hereditary molecule on Earth was not DNA. We actually think it was RNA. And we'll talk a lot more about RNA a little bit later on in the semester. But in order to make a piece of DNA, you have to prime it. And that's what the RNA primer does. And that's what primase, that's an enzyme, that's what it does. It lays down a little piece of RNA. Now, this is where I don't like this picture, and this is why I swapped it. But that's what would have happened right down here. Right down here. As that DNA polymerase was starting way back over here, the first thing that would be laid down would be a little bit of that primer by that primase, and then the DNA polymerase 3 can then attach to the end of that RNA primer and then run on down the strand. So, it's like priming a pump. You need a little bit of that RNA to get it started. Now, later on, DNA polymerase number 1 up here is going to go back and remove that RNA and put DNA in its place. So the RNA is only there temporarily to get everything going. And that's what the primase does, is the enzyme that attaches those RNA primers. So we get the primase, attaches the RNA primers, then the sliding clamp, and the DNA polymerase says, DNA polymerase 3 says, oh, okay, here's where we start. So that's what happened back here, and boom, we move on down the strand. Okay, now.
Are you going crazy for him,
Hmm?
He's going down the strand this way.
He's going down the strand from left to right. Yep, he's going down the strand from the left to right. Going down the strand, the original strand, remember which one is original? This is the original on the outside over here, and this is the original here. He's moving down that in the 3 to 5' direction. Now, that's a law of chemistry. Nothing us living critters can do about that. It's just the way DNA is. You have to move in that direction. And I've said this before, physics and chemistry and everything when it comes to biology, we're basically slaves to those laws of physics and chemistry. They were here long before we were here. The universe was doing physics and chemistry long before it was doing living things. So living things have to obey all the laws of physics and chemistry. Doesn't seem to be a big problem here, except for the other strand. Remember, it's anti-parallel. The other strand, you can't read from the 3 to the 5' direction because the other strand is anti-parallel. So you actually have to read it the other way. So what happens in this case is the RNA primer doesn't start at the end of the strand in this lagging strand. It starts further up. And then the DNA polymerase attaches, and then it attaches bases to it. And then what it does is it jumps back over itself. So basically the way it works is you read a piece of DNA, you make your other strand, you stop right here, you go back up, you read another piece of DNA, you stop right there, because you have to go in the same direction. You see, in another strand, we're copying from 3 to 5, right? Oh, I'm sorry, 5 to 3, 5 to 3, so we're going the wrong way. Trying to think of a good analogy to imagine what this would be like, but it's leapfrog is the only thing that comes to mind. Every time we create a piece of that DNA, then that DNA polymerase has to jump back up further upstream and read down to meet it, because it's got to follow in the same direction. So what it's doing is when it's reading a piece of DNA and then jumping back further up, it's still following the same path. It's still going in the right direction. I wish I could show with my hands. It can't move this way, right? It can't move. I'm looking at the top strand. The DNA polymerase cannot move consistently this way because it's moving in the wrong direction. It's going to move this way for a while, then it's going to jump back further stream and move down, then it's going to jump back and move down. All right, so to erase, the DNA polymerase would have started down here. It may have read those parts, so it would have been a primase with a little bit of primer, so we'd have read those parts. Then the DNA polymerase would jump up further. There'd be some more primase and another primer gets laid. But what you end up making are things called Okazaki fragments. It's actually named after a Japanese fellow who figured it out, who, interestingly enough, and just for the heck of it, I looked him up one time, he was a famous Japanese microbiologist or molecular biologist, but he was also a survivor of Hiroshima. He's actually there when we dropped the atomic bomb. So I think that's kind of interesting when you think about challenges in life that you have to get through. You know, this guy becomes a famous scientist, and his life started off, you know, fairly dramatically. All right, so everybody understand how that works? So we can read the leading strand straight across. The lagging strand takes a little more time. That's why it's called the lagging strand. And if we were to time this, the leading strand would get copied first, and the lagging strand would lag behind. It would take a little bit longer. DNA polymerase, again, is going to remove those pieces of RNA primer, because it all wants to be DNA. DNA ligase is going to stick those fragments together. Those Okazaki fragments, so you're getting a bunch of these Okazaki fragments, and the DNA ligase is going to actually bind those together.
Can you point to the Okazaki fragments? Those little bars coming?
Yeah, okay. That's a fragment. That's a fragment. See, there's a space in between here.
Yeah.
Right? So that DNA polymerase has already read this part, then it jumped up here, and now it's reading this part. So it's always jumping behind itself. Oh, these single-stranded binding proteins, just to kind of, what they do is when you unzip the DNA wants to go back together. It's like a magnet. It's hydrogen bonds that keep those bases together. The single-stranded binding proteins prevent them from coming back together. They keep it open long enough so that the enzyme can move through. Any questions on that? Okazaki fragments? Just don't like the way this picture is done. The leading strand is fine, but the lagging strand, just the way yeah. You guys got the idea, but I have a better picture of that that I'm going to switch that with. All right, things do happen, mistakes do happen when you copy DNA. Not everything's perfect, and there are enzymes that go along and help fix these things. Like, for example, we got a problem with that piece of DNA in the upper right that got a couple of bases that didn't bond properly. An enzyme known as nuclease comes along, cuts that stuff out, right? Gets rid of those bad pieces, and then division of labor, even among cells, DNA polymerase comes in and adds the new bases, fixes the pothole, if you will, and then DNA ligase comes along and glues everything together. Now, your body's ability to fix those things depends on how fast you add them and how fast they occur. So does it 100% always fix mistakes? Well, no, of course not. It doesn't. It does a pretty good job, but mistakes do get through. And sometimes those mistakes are good things, by the way. Sometimes those mutations are good things, but very often they're not. Anytime you monkey with the DNA, you got a pretty good chance you're going to screw it up. We deal with this here in Florida a lot with this problem that happens in our DNA because ultraviolet light, from the sun is very good at making these things called thymine dimers. If you look at that little piece of DNA down below, what it does is it causes those two red thymines to stick to each other and not stick to the adenines across. And that can lead to melanoma, skin cancer. That's where that comes from. Thymine dimers. Because if you look at that thymine dimer, if those two thymines are stuck together, that code is all messed up. You're not going to be able to read that code. Sometimes these changes occur by UV light or other physical agents, or sometimes they're spontaneous changes. As I said, even in prokaryotic cells who make clones of themselves, every time you copy DNA, it may not be exactly right every time. Nothing is. That's very, yeah, Penny says nothing is, and that's right. In life, in biology, it can't be perfect. That's right. Okay. How old do you want to live to be? Didn't we talk about this? Yeah, decent health. Want to live to be 100. They say that, the generation, not ours, Penny, unfortunately, but their generation has got a good chance they're going to live to be 100. And that's pretty good. As long as I'm in decent health, I'd like to be that. And we all know why we die. We die because we fall apart because of entropy, right? But let's take a look at a specific type of entropy that occurs. Now, up above, once again, is a leading and a lagging strand and so on and so forth. And what's going to happen based on the way those Okazaki fragments are formed, and if you look down here, when you're done making a copy of the DNA molecule, what you're going to have is you have the original strand, and on the end of the lagging strand is a little bit of RNA primer. Okay? Because you need a little bit of RNA primer because that's what starts the DNA polymerase. Now, that's at the very end of the strand. Now, this fragment right here of RNA primer, no big deal, because the DNA polymerase 1 is going to come along and replace that. But DNA polymerase 1 can't get rid of that last primer. The reason why is because DNA polymerase can't bind to that next to last base right there. All right, it's again a part of chemistry. So it can't get rid of that last primer and replace it with DNA. So that last primer falls off, but nothing's added to it. So you actually get a jagged end to the chromosome. So you got that chromosome where the one on the bottom is, what 5 bases longer than the one on top? And that's because that space was where the RNA primer was. So you can get rid of the RNA primer, but you can't replace it with anything. So you get molecules, DNA molecules, when they make copies, where one strand is longer than the other. Now, the next time that molecule replicates, since there's nothing on the other side here, essentially, you get a shorter molecule. And every time DNA replicates itself, every time the molecules get shorter and shorter and shorter, right? And you can actually see these shortened areas or these tips of the chromosomes are actually in this picture. They're called telomeres, and the little glowing part of what they're talking about, the telomeres. Now, if you're looking at that and you're thinking, well, that sucks, because what if that messes up a gene? Then you're on the right path of where I'm going with all of this. Telomeres is one of the reasons we get old. What are those little things called at the end of a shoelace? There's always a name for them. They always have that weird name. Here's your shoelace, and you got that plastic piece at the end. It starts with an A. It's always one of those crossword puzzle words that you can never figure out what it is. Ah, Courtney. How'd you know that? Courtney was an aglet. That is an aglet. How'd you know that? Huh? Hi, Jordan. What's Phineas and Ferb? that the tip of a shoelace was an aglet. What does an aglet do? What's its purpose? Yeah, I know when I was a kid, when I had tennis shoes, the aglets didn't last very long on my tennis shoes, and then my shoelaces were just, It protects the shoelace from unfraying, doesn't it? Yeah. Once you get rid of the aglet, like I said, your shoelaces become all frayed out and everything. So that's what the aglet does, right? Think about telomeres as being an aglet, all right? But it's getting shorter and shorter and shorter through time. The ends of your chromosomes, where all that DNA material is, have no genes. There's no genes there. There's adenines and guanines and cytosines and thymines and all those bases we talked about, but there's no genetic information there. It's filler, okay? It's not read by your body. It's again part of those silent gene or silent parts of the DNA. We can't really call them silent genes because the gene is expressed. It's pieces of your DNA that are just there. And the ends of your DNA have lots of those. And the reason why is because it protects the tips of the DNA. It protects the tips of the DNA. As you get older, your telomeres get shorter for the reason that we saw right here. It's because of the way the leading and the lagging strand. The lagging strand always ends up with that RNA primer that can't be replaced. So the molecules get shorter. Now, when you're young and you're cutting off the tips of those DNA molecules, it's no big deal because you're cutting through junk. There's nothing there. You're cutting off pieces of DNA that aren't going to be used. But as you get older, and those molecules are getting shorter and shorter and shorter every time you replicate. Now you're starting to get into the DNA that you use. Now you're starting to cut, that molecule is shortened enough now, so when this leading and lagging strand problem occurs and that molecule gets cut off, you're cutting off real genetic information. That's one of the ways, or one of the researchers into how we could prolong aging, is if we could figure out some way to stop those telomeres from forming. If we could stop the telomeres, not forming, from getting shorter, then we could probably, at least in one regard, prevent a lot of the aging process that occurs. Because you can imagine, as you cut into the molecule, into the good parts of the molecule, as it gets shorter, you start cutting into the genes that are necessary, and that can mess things up. Really interesting research being done out there. Telomeres have the ability to basically prolong our lives. For, I don't know, not indefinitely, but certainly for a longer period of time. So think about that. As you get older, and every time your DNA makes a copy, it's getting shorter and shorter and shorter.
What would be a reason for your DNA to make a copy?
Every time a cell, like during mitosis. Mm-hmm. Yeah, every time. Every time mitosis happens, every time meiosis happens, every time you copy DNA, you're going to copy it with the leading and the lagging strand. You're going to get that piece at the end that's going to basically nothing. It's okay when you're young, but when those things get really, really short, that's when it becomes a problem. All right, folks, last slide. We're a little bit early today, but that's okay. Up at the top, what we're looking at a DNA template, excuse me, a DNA template strand that's 3, 6, 9, 12, 15 base pairs long. There's the complementary strand. They've labeled the 3 and the 5 to remind you it's anti-parallel. There's the piece of messenger RNA in the red. We'll talk more about how the messenger RNA comes along, but the messenger RNA is what relays the code to the ribosomes. You remember the ribosomes is where the proteins are being made. The DNA is in the nucleus. So the messenger RNA takes the message from the DNA, goes out to a ribosome and says, here's the recipe to make this protein. Here's how you make it. And so, for example, we're making a protein here that's, 4 amino acids long. We got methionine, lysine, phenylalanine, and glycine. Very simple protein, all right? Wild type up above means normal, the way it typically is, okay? Without being mutated. That's what they mean by wild type. So let's look at the ones on the left-hand side first and see what they could do. If we have a nucleotide pair substitution, so for example, where that 4th guanine over up at the wild type, this one right here with the arrow that I drew at the very top, if that is replaced with an adenine, then of course on the other side it's going to be a thymine. And then we'll talk in a minute how that, or we'll talk later on how that ends up in the messenger RNA. But because of where that substitution and mutation occurred, that does not change the amino acid that's there. So you still got methionine, lysine, phenylalanine, and glycine. So technically, you had a DNA mutation, right? But it does not show up. It's silent, like you see in the box there. It's a silent mutation. As long as you end up with the same amino acid, it doesn't matter if the DNA is changed. And in subsequent chapters, we'll see that Mother Nature has been very kind about that. She builds a lot of fudge factor into the code. So long as you get the code relatively close, you're going to get the right DNA molecule, because nothing's perfect in life, right? Nothing's perfect. Nothing happens perfectly. However, if you change one of the bases in a way that changes the amino acid, like you see the one down below, it's thymine instead of a cytosine, that means you get a different amino acid. And that's what we call a missense. Now, different amino acid. Is that bad to have a different amino acid in that protein?
No.
Might not be bad. Penny says it's not bad, and she's right. In some cases, it might not be bad. Let's imagine that glycine and serine were both hydrophobic amino acids. Remember, there were hydrophobic ones and hydrophilic ones, and there were acidic ones and basic ones. So let's say it's the same basic type. It might get the protein to fold pretty close to the original shape that it's supposed to be. And enzymes are proteins, and their active sites accommodate the substrate. So maybe sometimes a little bump here and there is not such a big deal. What if that amino acid, serine, was, let's say glycine was hydrophilic, and say serine was hydrophobic? That's totally going to change the way the protein folds, right? And the shape of a protein is very important. So that could, that missense there could have no effect or very little effect, all the way up to a major effect. For example, in sickle cell anemia, folks with sickle cell anemia get the sickle-shaped red blood cells because of one letter wrong. One of their bases is wrong, that's it. But that one letter that's wrong ends up coding for an amino acid that totally changes the structure of the protein. It's amazing when you think about it that way. One letter difference. Sometimes what happens is a mutation, like you see on the bottom, occurs very early. Some of those letters indicate stop. And what stop means is it tells the ribosome, this is where you stop. Because many times a piece of messenger RNA, the stuff in the red, will actually code for many, many, many proteins. And it's sort of like on CD players. It's the track. It's where the tracks break. So you have stop codons that says, all right, this is protein number 1. Like up at the wild type, methionine, lysine, phenylalanine, glycine, stop, there's your protein. And then after that, you might actually start another protein, but this is the stop. If a mutation occurs that creates a stop signal right after the start, you get methionine, for example, stop. That makes nothing. That's a nonsense. That doesn't make the protein. Methionine is an amino acid, it's not a protein. So you've done nothing there. You don't create the protein product. Maybe being lactose intolerant. Maybe if you were born lactose intolerant, if it's genetic, you don't make the lactase enzyme. And the lactase enzyme is a protein. And maybe that's why. Just a possibility is because there's a mistake where when you go to your chromosomes to read about how to make a lactase enzyme, some mistake has occurred and it starts with the first one and the next thing says stop, so it doesn't make anything at all. That would cause problems, right? So between these three, the silent would have no effect. The missense, depending on the amino acid, maybe a little effect, maybe a lot of effect, maybe a middle effect, and then a nonsense would have a tremendous effect because you're not making anything at all. So you can do that with a nuclear pair or nucleotide pair substitution. You can do it with a nucleotide pair insertion or a deletion. This is what we call a frame shift error. If you think about it, or if you don't think about it, but if you look at it, what we were doing, and again, this is going to be more apparent in subsequent chapters, when the messenger RNA in the ribosome is read, when the ribosome says, okay, what's the recipe for that protein, and the code says methionine, it reads those letters 3 at a time. The ribosome reads AUG. There's a lot of AUGs on the page, so I'm at this one right here. The ribosome reads AUG, and it knows in code that's methionine. And if we go back up to the top, just to kind of keep going, AAG, that codes for lysine, UUU for phenylalanine, right? So if you have a situation where a U was put in there, right? Or actually an extra A. And in the messenger RNA ends up forming an extra U. So that was shoved in there, and it shoved everything further down to the right. All the letters are there, but now we got an extra one. Well, because you put that extra one in there, you get the same thing as the nonsense from the lower left. You get methionine, then you get a stop, so you don't get the protein at all. That's a problem. If you had a missing one, like in the second one, oh, these are really bad. If you have a missing one, like in the second one, take a look at it. Every spot downstream of where that one was missing, or an insertion would do the same thing. The insertion they just showed you ended up forming a stop at the very beginning. But an insertion or a deletion can do this. Every spot downstream from the insertion and the deletion is going to be screwed up. Everything. It's like misbuttoning a button shirt. You know, you button the top button, you get it screwed up, you get to the bottom, you're like, oh man. You can't fix it unless you totally unbutton the shirt and start all over again. So you see what's happening here. We get methionine, lysine, and of course the ribosome doesn't know that a base is missing or a base has been added, and it still reads them 3 at a time. And so the U, which would have gave it phenylalanine, is now UU. Take the G for the next one. UUG is leucine, and then GCU is alanine, and then it keeps going. These kinds of frame shift mutations are particularly detrimental because, like I said, if a piece of messenger RNA is coding for 30 proteins, and this mess up happens at the beginning, you screwed up all the stop codons down the road. Everything is messed up. Frame shift mutations can be particularly damaging because you shifted the frame at which this is read. Finally, the other thing that can happen is instead of just a pair of nucleotides being deleted, you can actually miss an entire group of 3. So, for example, this one, the TTC is missing out of the DNA. So that means down on the messenger RNA, you miss an amino acid. So methionine, phenylalanine, supposed to be a lysine in between there. You don't have that lysine amino acid. Now, what kind of damage does that cause? Again, it depends on the protein, depends on the severity of the mistake. You know, it could be from something very simple to something rather dramatic, obviously. All right, folks, that's the last slide. That's all I'm going to do today. I know we're stopping a little bit early, but we still got some time. We'll do 15 and 16 as we go through the next few weeks. And we should have plenty. You got your exam on Tuesday. Don't forget. We won't have lab on Tuesday. There is no lab, but I would like us to stick around a little bit after the exam and maybe talk about the lab practical, kind of go over some of the ideas, maybe and work some more on those genetics problems too. So, and again, hold off on the genetics problems that I handed out. We'll work through those in class, but I got to show you what the mistakes are first, okay? In fact, that'll be a good class thing that we can all do together. Does anybody have any questions for me? Miss Penny, you okay? Anybody else? All right, folks, I'm going to stop the recording. You all have a good weekend.